Hello everyone, I am back with my new video of biology today. Okay, so today's chapter is cell, the unit of life. We have previously discussed a lot of things, and today also we are going to discuss some other things. So before starting our today's topic, let us first recapitulate everything which we have discussed previously. So who was the first person to see the cell? It was Robert Hooke. Okay, he observed a cell wall basically of the which plant one? Spanish oak tree, and its scientific name is Quercus suberg. He took the cork of it, cut it into thin slices, observed it into under the microscope, and then saw beehive like structure, named it as cella, and later cell was derived. He basically saw the cell wall of that from that tree. Okay. Next, who was the first scientist to see a living cell? It was Anton van Leeuwenhoek. In from where? From the rainwater. He saw the microbes moving here and there and named it as animalcules. Then who was the then comes cell theory. Cell theory was basically given by three scientists. Okay. First one, there were 2 points in it. First point was given by Matthias Schleiden and Theodor Schwann. Matthias Schleiden in 1838, he was a German botanist. He studied about plants. Okay, he found that all plants are made up of cells and its products. Then in 1839, British zoologist named Theodor Schwann saw that all plants as well as animals were made up of cells and its products. So they gave the first point in the cell theory that all living organisms are made up of cells and its products. Now second point was given by Rudolf Virchow. In 1855 he came and said omnicellula e cellula, which meant all cells arise from pre-existing cells. So these were the 2 points by which cell theory was completed. So Rudolf Virchow completed the cell theory, modified the cell theory, and gave the cell lineage theory. Now next comes is a typical plant cell and typical animal cell. Typical plant cell given in NCERT is onion cell, and typical animal cell given is human cheek cell. So we observed the differences between them in the previous videos. So first difference was cell wall was present in which? Plant cell and absent in animal cell. Similarly, plastid or chloroplast is present in plant cell and absent in animal cell. Large vacuole is present in plant cell and absent in animal cell. Then centriole is present in animal cell and absent in plant cell. Okay. Then eukaryote, eu means true and karyon means nucleus. So eukaryote basically means true nucleus. Okay. Then we also studied about the bacterial cell. It is a prokaryotic cell. Pro means primitive and karyon again means nucleus. It is a primitive type of nucleus. So bacteria, we discovered about the structures of bacteria. Basically the outermost structure is called glycocalyx. Glycocalyx is further made up of 2 parts. Now the hard part, the solid part of glycocalyx is called as capsule, and the another part is known as slime layer. Okay. So work of capsule is to protect bacterial cell from RBC, and it also helps in colony formation. Now work of slime layer is to is attachment, to get bacterial cell attached to something, and another is prevent it from drying out. Okay. Now then comes cell membrane. Cell membrane is made up of what? Peptidoglycan, and there is also a periplasmic space present in between, and it contains what? Digestive enzymes. Okay. It works as the lysosomes of eukaryotic cells. So it has been compared with it. Then comes mesosomes. Mesosomes are basically invagination structures of the cell membrane. Okay. Its work is respiration basically, but it also helps in DNA replication, cell division, protein synthesis, enzyme synthesis, cell wall material synthesis, etc., etc. Work of mesosome has been compared to mitochondria of eukaryotic cells. Okay. Eukaryotic mitochondria's main function is respiration. Okay. And it also helps in what? ATP generation. So Next thing comes is differences. We studied the differences also. Now bacterial cell also has longer pili and shorter pili. Longer pili is also known as sex pili, which helps in conjugation, and shorter pili is also known as somatic pili, which helps in attachment. Okay, their pili are made up of pillin protein. Now next is flagella. Flagella is made up of flagellin protein in bacterial cells, and it helps in locomotion, or can say movement. Okay, then they have double stranded circular DNA, and plasmid also. Plasmid contains characteristics like phenotypic characteristics like what do we say, fertility and resistance to antibiotics, or can say resistance to WBCs also. Okay, now next thing comes is ribosomes. Bacterial cell have 70S ribosomes, whereas eukaryotic cells have 70S and 80S, both type of ribosomes. Okay, now next thing comes is we studied the differences also. Basically there were 8 types of differences. First is nucleus, then respiratory enzymes, then comes vacuole, then comes chromosomes, then also cyclosis, then also ploidy and flagella, and also compartmentalization. Compartmentalization is absent in bacterial cells, or can say prokaryotic cells, because they lack membrane bound cell organelles, whereas compartmentalization is present in eukaryotic cells. Okay, now that were the differences. Now today we are going to study about H.C. Gram scientist. Okay, he basically discovered the Gram staining technique. Okay, he colored the bacterial cells, and the bacteria which were colored were called as Gram positive bacteria, and bacteria which were not were called as Gram negative bacteria. Okay, after that I will let you know the size of some cells, like RBC are 7 micrometers long, then comes WBC, which are not WBC, eukaryotic cells, which are 10 to 20 micrometers, then comes bacterial cell, which is 3 to 5 micrometers, then mycoplasma, which is 0.3 micrometer, then PPLO, pleuropneumonia like organisms, which is 0.1 micrometer, then largest isolated cell is an ostrich egg cell, then also longest cell is the nerve cell. Okay, cells size and shape vary according to their function. Different cells have different shape as per their function, like RBCs have round and biconcave because they have to move through thin vessels, so they are small, round and biconcave. Then next one is neuron or nerve cell. It is long and branched. Then is tracheids. They are elongated because transportation of water and minerals occur through tracheids and vessels. Okay, then next is WBCs. They have amoeboid like shape because they have to stretch their body and legs to engulf the bacteria and protect us from pathogens. Okay, next one is mesophyll cells. They are basically round and oval in shape, and they contain chloroplasts which are required for photosynthesis. Then columnar epithelial tissues, or basically the cells present there are long and narrow. Okay, now then comes cell membrane. Cell membrane is also known as biomembrane, plasma membrane, or can say unit membrane also, or plasma lamella. Okay, these names are important as per the perspective of NEET also. Okay, so let us study about the structure, physical and chemical properties. Physical properties goes like this. They are thin in structure, they are living. Since cell membrane is present in all over the cells found in the world, as we have discussed, for being a living cell, cell must contain cytoplasm, cell membrane and nucleus. So cell membrane is found in all the cells, even in prokaryotic and eukaryotic cell. Okay, so they are universal. Next comes is semi-permeable and also selectively permeable. They do not allow all the materials to pass through them, but allow only specific materials to go in and out. Okay. Now comes the chemical properties, like they are made up of phospholipids, protein, carbohydrates, etc. Okay. So in RBC, basically lipid content is 40%, protein content is 52%, and carbohydrate content is 8 to 9%. Okay. Now let us see their structure. Basically, details about cell membrane was found after 1950s, but we have discussed that cell theory was already given in completed basically in 1855 around. So why does it took a long time? It was just because of the wars going on. So people were basically finding safe spots to protect themselves from the war, so they were not getting enough time to do research and all these things about these. Okay, let us see the structure and know about it one by one. This is basically the structure given by the first model known as sandwich model. Yeah, it's a funny name, sandwich model, also known as trilamellar model, because it has 3 layers: one protein layer, then lipids bilayer, then again a protein layer. So trilamellar layer, then also known as PLLP model. It was given by the scientists Danelli and Davison. Okay, so this layer is protein layer, which has a length of about 20 angstrom. 1 angstrom is basically equal to 10 to the power minus 10 meters. Okay, so it is 20 angstrom. Then comes lipid bilayer, which is around 35 angstrom, and then again a protein layer, which is of 20 angstrom. So basically they have given a range of cell membrane from 75 angstrom to 100 angstrom it can be. Now there are some bonds also present between them, which are ionic bonds and hydrogen bonds, and also between lipid layers van der Waals forces are also there, and between proteins also there are pores present in it, which are 0.3 millimeter wide. Okay. Now this lipid also has 2 parts, head part and tail part. This above round part is the head part and this one is the tail part. Now tail is non-polar in nature, so it cannot dissolve in water, and it is hydrophobic, so it does not like water. And head is hydrophilic, so it is polar in nature and is hydrophilic. It loves water. Okay, so this is the basic structure of cell membrane. We will discuss about it in detail in the coming videos. Okay, that's all for today's cell. Thank you.